All right, good afternoon, everybody. It is 12 o'clock on a Friday, which means it's time for the novel coronavirus update for clinicians. I am Dr. Michelle Fiskus, and with me today is Dr. Kara Levinson, who is the Deputy Director from Laboratory Services here at Tennessee Department of Health and the Tennessee State Public Health Lab. We will have a few Mentimeter questions today, so if you'll go to www.menti.com and use the code 942863, you can answer our polls and move through those at your own pace. So I invite you to do that. It gives us just a little bit of feedback about these webinars, what you want to hear about, and kind of what you all are dealing with out there on the front lines. So I'm Dr. Michelle Fiskus. I'm the Medical Director of the Vaccine Preventable Diseases and Immunization Program here at TDH. And as I mentioned, Dr. Kara Levinson is with me today to answer some questions about laboratory testing. We've gotten the feedback from those of you who've been on the webinars previously that you wanted to know a lot about treatments and vaccine updates and testing, and so that's what we're going to talk about today. A little bit of housekeeping. So as we always say, the guidance that we give is rapidly changing. This is a constantly and rapidly evolving situation. So anything that we say today might be outdated tomorrow, but it's the best information we have today at 12 o'clock on April 17th. We do post the slides and the recording following the call, and that will be up on the Tennessee tn.gov/health coronavirus website under Healthcare Providers tab. All of your lines are muted, but you can ask questions in the Q&A box, and Peter will put those up on the screen for us to answer. We usually have about 30 minutes at the end of the presentation here to get to those questions. We'll answer as many of those as we can during the call, and the rest will come out onto the ongoing written Q&A that we have under that same Healthcare Providers tab at the tn.gov/health coronavirus website. So again, go to www.menti.com, enter 942863, and you can participate in our polls. So the update for right now with our response here at TDH is that we are 43 days since our first case, and we have been in our incident command structure here for 93 days. We still have our clinician call-in line during the day that's answered from 8 to 8 Central Time, 7 days a week, and that number is 615-741-7247. We also have one of our CDEP physicians on call overnight for any kind of public health emergencies. Those still happen, like rabies bites and contaminations and carbon monoxide poisonings, and we today for you as well. Soon, I know the first wave of money has gone out to some healthcare workers. I know that the second wave that's coming out is specifically also coming out to pediatricians and pediatric hospitals. So hopefully you'll start to see those funds showing up in your bank accounts to help with some relief from that. And then, have you ever attended these Friday updates before? So it looks like the majority of folks have been here at least, well, more than once before, and but we've got a growing number of folks that are joining us for the first time, so that's really cool. And we do these every Friday at noon until they tell us we can't anymore. For anyone who's feeling really stressed, I want to remind you about the free confidential telehealth line for physicians, and that is staffed by volunteer psychiatrists from across the United States. Specifically to help with talking through stress around this pandemic. So you can go to the site that's listed on the screen. They're there from 8 in the morning until midnight Eastern Time, 7 days a week. If you feel like it might be beneficial to talk to someone, you might give those guys a call. So this is the national update as of yesterday. So the U.S. map is up on the screen, and you can see that we still have the largest number of cases on the coasts, but then also in the southeastern part of the United States and then in the Midwest. And it's interesting, I was starting to put up a similar graph from April 1st to compare to this one, but they had to change the scale of the graph to where the darkest color went from 5,000 or more cases to now 10,000 or more cases, because pretty much the whole map was turning into the darkest color of orange. So I couldn't put a comparison up here, but you can see that New York is still very much impacted, but then we have a growing number of states that have crossed the 500,000 case mark at this point. Down below is the national epi curve as of yesterday, 2 days ago, at 632,000 cases. That's about 0.2% of the United States population that's been identified as COVID-19 positive. About 0.1% of the population has been tested, and about 21,000 new cases are being reported each day in the United States. We post our updates every day at 2 o'clock Central Time here on the tennessee.gov/health coronavirus website. And as of yesterday, we had 6,262 cases, 141 deaths, and we've got just about every county in the state now that has declared cases. I think there's still 4 or 5 that do not yet have cases. The leader here is Shelby County with 1,492 cases, then Davidson County with about 1,300, and then the counties surrounding Davidson and Knoxville have the next highest numbers of cases. And then our deaths in Tennessee, 82% of those deaths are in individuals who are 61 years of age and older. We have had 1 pediatric death and a fairly low number of folks under the age of 40 that have had fatal cases of COVID-19 as of yesterday. We also like to give you the update, and I forgot to update the date at the top of this, but this is as of yesterday. We have 35% of the hospital floor beds that are available, 32% of the unit beds, about 52% of the isolation rooms, and we still have 75% of the adult ventilators in the state that are available at this point as of yesterday. And these numbers have stayed pretty consistent from the 1st of April, with that floor and ICU bed number hovering in the low to mid-30s, and the ventilators hanging out about 70 to 75% available. So we're still in pretty good shape as far as what the hospitals are reporting to us. This is an influenza-like illness chart that came out a couple of days ago from the National Influenza-like Surveillance System. And you can see that where we have practices that are doing surveillance work across the United States, you can see what their influenza-like illness numbers are looking like. These are people who've presented to offices with cough and fever as their chief complaints. You can see in Tennessee, in the Shelby and Nashville-Davidson areas, those are where we still have our highest numbers. But if you would like to assist with helping us collect this surveillance information, I'll remind you about the Sentinel Provider Network. We have been fortunate enough to have some of you who've listened to this webinar volunteer to be Sentinel Provider Network providers, and we're really grateful for that, because this helps us understand what flu and novel viruses and RSV and other types of respiratory illnesses are looking like across the state of Tennessee. If you're interested in helping us out with that network, you can email cassandra.jones@tn.gov, and she will be more than happy to walk you through the process of becoming one of our SPN providers. A reminder that the health departments have drive-through testing capability, and this Saturday and Sunday there's a greatly expanded capability across 33 departments of health across the state. You can go to this site, which is actually on the governor's page, so tn.gov, click the red bar at the top of the page and scroll down to that blue box that says remote assessment sites. There's a map there where you can click on your county and find out where the testing sites are in your county that are closest to you, and also get the information about when those are available. For tomorrow, here's a list of the counties that will have drive-through testing and where those are located. So some are at high schools, some are at churches, some are at health departments. In Madison County, it's at Jackson Fairgrounds. Most of these places are operating at least 9 to 12 tomorrow, but some of them have more expanded hours. So there's this list. This is also posted to that remote assessment site. Here's some other counties that will have testing available tomorrow, generally from 9 to noon. Van Buren, I'll mention, is open 3 to 6 tomorrow and not in the morning. And then on Sunday, there are several sites that are also open across the state. It's a smaller list. I think there's 11 counties open tomorrow with testing. So these are drive-through testing. You don't have to have any symptoms to get tested. If you feel like you need a test or you have a patient that needs a test and they want to go to one of these sites over the course of the weekend, you can send your patients there to get tested at no charge to them. I want to talk a little bit about where we are with research on treatment for COVID-19 at this point. This is a JAMA Network publication that came out just a couple of days ago. This really cool illustration here of where these different medications might be influencing the activity of the SARS-CoV-2 virus. So first, there's been a lot of interest around chloroquine and hydroxychloroquine, which are antimalarial medications that are also used to treat chronic inflammatory diseases like lupus and rheumatoid arthritis. It appears that these medications may block the virus's entry into the host cells and also maybe attenuate the cytokine storm that may happen with people who are severely ill from COVID-19. But there's still no high-quality data that exists to show that these medications are of any benefit in COVID-19. In China, there was a study of 100 patients that showed some reduced disease progression, but that was not validated. There was a smaller study in China of 30 patients that were randomized, and the standard care group that did not receive hydrochloroquine actually did better than the group that received the medication and clearance of the virus. And then France had an open-label study of 36 patients, which showed that they did have improved virus clearance, but 6 patients had to be removed from the study because of the critical nature of their illness and intolerance of the medication. So dosing has still not been determined for these medications, and you have to consider the serious adverse events that are possible, like QT, prolongation, retinopathy, hypoglycemia. So it is still not recommended that these medications be used in standard practice to prevent or treat COVID-19 illness at this point. There are a few antiretrovirals that have been looked at. These are HIV treatments that look to have in vitro activity against the novel coronaviruses. There have been a few retrospective studies looking at SARS outcomes with people who were already receiving these antiretroviral medications. But there was an open-label randomized control trial of 199 COVID-19 patients that showed no difference in outcome between the treatment and control groups. And there's significant concern about especially the hepatotoxicity of these medications, especially since COVID-19 itself tends to cause at least a transient elevation in liver enzymes, that adding medications to COVID-19 patients that cause hepatotoxicity is probably not a good idea. There has been talk about alpha or beta interferon as a treatment. There are currently still no animal or human data to support the use of any interferons as a treatment or prevention for this disease. Ribavirin has been looked at. That's a guanine analog that inhibits the viral RNA-dependent RNA polymerase. There were some studies done with SARS patients where they showed no conclusive evidence that ribavirin was beneficial, and 4 studies actually showed demonstrable harm from the use of ribavirin. There's no significant data around using this in coronavirus, and again, there's significant dose-dependent hepatotoxicity. About 60% of the patients who were treated with it got hemolytic anemia, and it's a known teratogen, so it's not safe to use in anyone who is or may become pregnant. There's an anti-helminthic medication that has had some broad antiviral activity in vitro. against MERS and SARS-CoV-2, but still has not been used in any specific in vivo studies for SARS-CoV-2, but that might be one that that we get some more information on going forward. Antiviral medications like oseltamivir, Tamiflu, has been looked at and has deemed to have no role in the management of COVID-19, so should not be used. Tamiflu can also impact the outcome of some of the serologic testing for COVID-19, so if you have someone who's on Tamiflu and then you try to test them, you may not get the reliable results from those tests because of the Tamiflu. And then I have trouble saying these names every time. Umilfenavir, Arbidol, is a medication that is being looked at in China. They have a non-randomized trial of 67 COVID-19 patients where it showed significant mortality reduction, 0% mortality in the treatment group versus 16% in the untreated group. That medication is currently approved in China to be used in patients who are critically ill with COVID-19. Remdesivir is another medication that's being looked at seriously here. That's a monophosphate prodrug that was developed to fight Ebola and does appear to have broad-spectrum potent anti-COVID-2 activity, and it's well tolerated, and although it has a side effect of transaminitis, it seems to be reversible. There is a current NIH randomized controlled trial going on right now versus supportive care, and it can, while it's not FDA approved yet for the treatment of COVID-19, it can be obtained via compassionate use and emergency use through expanded access if there's no clinical trial access where you are, and individual compassionate use for pregnant women and children who have severe life-threatening disease. So I expect we'll hear more about remdesivir over the coming months. Adjunctive therapies like corticosteroids have really shown some increase in mortality and adverse outcomes with use. So again, there's caution against using corticosteroids unless they're being used as part of a random control study or there is a concomitant compelling indication. So you have someone with COPD who needs the steroids for their COPD or their asthma exacerbation, and they have COVID-19, you may feel like there's not a choice but to use corticosteroids, but they should not be used in patients who just seem to have COVID-19 symptoms. CDC and WHO have both published warnings about using systemic steroids in patients with COVID-19 infections. And then there have been a couple of adjunctive therapies looked at. Some are monoclonal antibody therapies that can be used to potentially dampen the cytokine storm that we see in severely ill patients. There's been a case series with 21 patients with COVID-19 that showed a 91% clinical improvement. There are some randomized controlled trials going on in China, and China has now included an anti-IL-6 monoclonal antibody as part of their national treatment guideline. There are also clinical trials going on a couple of other monoclonal antibody type biologic products. And then convalescent plasma, a lot of folks have been interested in that. There's a 2009 study of critically ill H1N1 patients where there was a reduction in mortality to 20% versus 54.8% in the control group. In theory, convalescent plasma infusions would be most beneficial around day 7 to 10 of the infection, where there is high viral replication but still low innate antibody production. And there is a multi-centered study going on in the United States right now looking at convalescent plasma in severely ill COVID-19 patients. As far as vaccine updates, there are 6 players right now in the vaccine market. One is from Moderna Therapeutics, which is a United States company. That's an mRNA vaccine. These have never been used in humans, and in this particular vaccine, the animal studies have been skipped to try to move it to phase 1 trials faster. There's a phase 1 trial going on through Kaiser Permanente in Seattle, Washington, with the expectation that that phase 1 will end in June 2021 and then move to phase 2 clinical trials. There's a vaccine that's been developed in China that's a viral vector vaccine that's very similar to the Ebola vaccine that's now in phase 2 clinical trials. It's in a phase 1 clinical trial in Hubei province in China with an anticipated phase 1 end date of December of this year. They expect to have all of their testing done, but not until 2022. And then there's a vaccine out of the University of Oxford in England that's doing a combined phase 1 and 2 trial together at the same time. This is an inactivated viral vector that's derived from chimpanzees and funded by the UK government, and they anticipate that this vaccine might be ready for release in May of 2021. And then there are 3 vaccines of those 6 that are still in preclinical phase. One is a Pfizer product that's in collaboration with BioNTech. This is also an mRNA-based vaccine similar to the first one I mentioned, and they expect that they may have phase 1 trials sometime this month in the United States and Germany. There's an Inovio Pharmaceuticals product. This one is really interesting. It's an activation immunotherapy vaccine that delivers DNA into the host cell with a handheld smart device. And then that DNA is translated into proteins that activate the immune system to create a targeted response against the COVID-2 virus. They're expecting to start their clinical phase 1 trial in April in this month as well. And then there's a recombinant DNA vaccine from Sanofi in collaboration with Health and Human Services. That is a recombinant DNA vaccine that encodes for the COVID-19 surface protein, like many of the other Sanofi-developed vaccines that we see. That one is also expected to go into phase 1 trials in April of 2020. So I think that the take-home message of all of this is that we are still probably at least a year away at a very optimistic point of view from having a vaccine that can be used in the population. I was listening to a webinar from Dr. Paul Offit out of CHOP a couple of days ago. He really thinks we're looking at more like 2 years to get a vaccine out. But it's unlikely that we're going to see anything in the next 12 months. And I'm going to turn this over to Dr. Levinson, who's going to talk a little bit about testing and what's going on with that.
All right. Good afternoon. This is Dr. Levinson, and as we shift gears towards testing techniques, we wanted to first share this instructional video. From the laboratory perspective, the quality of the specimen collected really impacts the quality of the results we can then provide you. And so the recommended specimen type is still the nasopharyngeal swab or NP swab. And so we wanted to share an instructional video on how to best collect that specimen in the hopes that you can get the best result possible. So you can click on that link there. Moving next to the prioritization of testing within the state public health laboratory. There's a variety of labs in the state that are currently doing COVID-19 testing, and we're really reserving a lot of the general diagnostic testing to be served by the private laboratories throughout the state, and reserving the state public health lab for those high-risk, high-priority specimens. So those include direct contact with a confirmed COVID case, travel to a high-risk area, whether someone is pregnant or immunocompromised, they have a high-risk occupation like a healthcare worker, whether they have severe pneumonia or ARDS, congregate care or incarceration or lack of insurance. These are all factors that would help influence whether or not a sample is taken to the public health lab or tested elsewhere. And so testing with the state public health lab has to be approved in advance by the Tennessee Department of Health, and we assign a PUI number, and that helps expedite getting the sample to the public health lab and getting your result as quickly as possible. Next is about testing for uninsured patients. We do offer testing for those that don't have insurance. Essentially, the process to do that is to confirm the patient's uninsured status, administer the COVID-19 test when it's clinically indicated, send the test samples identified as uninsured patients to contracted labs by your normal means, making sure you indicate that they're uninsured, and the lab does the testing, reports the result to the clinic, and then invoices the clinic for the tests, and those are submitted by the clinic with an invoice net due in 15 days to the Tennessee Department of Health, supported by confirmation of a patient's uninsured status and an invoice from the laboratory. So the clinic will send those electronically to Valerie Oliver at TDH. Her email is shown here on the slide. And essentially, TDH will process that invoice for payment to the clinic within 15 days at no more than $70 per test. And so the clinic will then process the original invoice from the lab for payment with the funds received from TDH. So there is a mechanism in place to be able to test patients even if they don't have insurance. Next, I wanted to shift gears and talk about testing. Serological tests have been the hot topic, at least the past couple days. So I wanted to kind of focus on what's new as it changes by the hour and certainly by the day. Really what we're looking at here, I wanted to highlight there are 3, and as of about an hour ago, now 4, serological tests that have received an emergency use authorization or EUA from the FDA. And the first 3 are highlighted here. I think the Cellex test was mentioned in last Friday's webinar. And they're all antibody tests. They're all pretty rapid, like certainly within an hour. And most of them look for both IgM as well as IgG antibodies. And they are all a little different from each other. So for these 3, the first one, Cellex, is a lateral flow assay. And they all take a variety of specimen types. So for this one, it can take serum, plasma, or whole blood, but notably not a finger prick. So it does require venous whole blood. And it can be read by the person performing the test visually, and you get results within 15 to 20 minutes. Alternatively, there's the ChemBio diagnostic system that just received their EUA. This also takes about 20 minutes. Notably, it can take venous or finger stick blood in addition to serum or plasma, but it also requires a DPP microreader to interpret the results. So you can't visually look at the card, even though it looks like you can. You're not supposed to. You have to have a reader, another piece of instrumentation, to get the result for this test. And then finally, there's the ortho clinical diagnostics test. This is more of an ELISA, so it's not a lateral flow assay. It takes a little bit longer, but you can get results on your first sample within 48 minutes. It only uses serum and plasma, and it kind of works like many sandwich ELISAs where you have a spike protein antigen at the base of the well, you add your serum or plasma, and then there's another antigen added, and you read the luminescence. Also, that requires another reader to get the test result. So Cellex is the only one that you can read visually. The other 2 require additional equipment to be able to do the testing. And one thing I wanted to note, all 3 of these tests require they be performed by laboratories certified by CLIA to perform moderate or high complexity testing. These are not designed to be point of care testing without that specific CLIA certificate. And I wanted to touch a little more on serological testing, kind of what we're learning as we go, what the current questions are, and kind of what we're working on. In a nutshell, we have far more questions than we have answers. One thing that's been mentioned in a variety of studies is the quickness of IgG being detectable compared to other infectious agents where you typically see IgM first and then you see IgG afterwards. Generally speaking, what people have found is that you see IgG pretty quickly, pretty much at the same time as you can detect IgM. It's very rare to have an IgM positive, IgG negative result. Not impossible, but it's one thing we're consistently seeing across the board as more and more serological tests are performed. So that's one thing we're starting to see. The questions we still have, and this is really critical to those that are thinking about implementing or using serological tests, is we really don't know a lot about cross-reactivity, whether it's with MERS, SARS-1, or other human coronaviruses, especially those seasonal ones. Those are things, and depending on the test, it really varies as to whether the manufacturer even looked for cross-reactivity amongst those other seasonal coronaviruses. We also don't know a lot about neutralizing antibodies. So if someone does get COVID-19, we don't know much about the immune response that they're mounting, how long they're protected, and the types of antibodies that they mount in response, and kind of how many of what proportion are neutralizing versus the other kinds of antibodies you can produce. So we really don't know a whole lot about how well someone is protected even after they get COVID-19. We know we can detect IgM and IgG antibodies, but we don't really know what that means yet. So those are the questions that are currently being looked at, and as more of these tests are put into place, hopefully we'll learn more soon. Moving forward, I wanted to touch there are now 3 serological tests that have the FDA EUA, and there's one added today from Mount Sinai. This isn't so much a standalone test as it is them offering ELISA-based serological testing, but so now there are 4 options in terms of serology. But we know a lot of companies are working to get their EUAs very, very soon, and these are bigger companies that can offer more high-throughput testing. One caveat to the test that I showed you before is you can only do 1 at a time on 1 patient, where some of these companies we know are working on much higher throughput. So those should be coming out in days to the next week or 2. So be on the lookout for that. Topics that are currently being discussed on the laboratory side is coming up with guidance on what to do when you get a serological test result, how to interpret that, and also testing algorithms are in discussion, and how do you balance molecular testing and serological testing, and one should inform the other, that kind of thing. So these are all practices and things we're thinking about as we go, as more of this comes on board. And then finally, I wanted to mention some seroprevalence studies are already underway. The CDC has a serological assay that they're employing for this. They're starting in areas of the country that are kind of hot spots, so certainly on the east and west coast, but they're looking at areas that have high cases of coronavirus. And really, they're not being used necessarily for individuals that want to know if they've been exposed to COVID, but it's really to get that populational data to figure out who has been exposed, whether or not they were symptomatic, and whether or not there's any kind of demographic information that could help inform who was more or less likely to get infected. So that's currently underway, and just as kind of a fun note, there's other seroprevalence studies happening, certainly, but also the Major League Baseball group is also participating in a seroprevalence study with a research group out in California, and they are working with not just baseball players, but members of their family as well as other people involved with the group, even down to ushers and people in stadiums and things like that. They're really utilizing MLB as a way to kind of get a subset and a cross-section of the population to estimate risk and exposure to coronavirus. So I put those 2 links up on there. The last one's kind of a fun read. But we're really using the seroprevalence and these serological assays to kind of inform at a populational level. They're really not recommended for diagnostic use at this point in time.
That's great information. So I have a question for you. So is the thought that because the IgG and IgM are being detected at the same time that maybe that is from cross-reactivity with other coronavirus antibodies?
So we don't know is the short answer. We're looking into that. The challenge is finding serum that have known other coronaviruses, especially recent ones. I would imagine the prevalence of SARS-1 and MERS is pretty low, but we know seasonally you see human coronaviruses circulating. So finding samples of that we can then check these is really what we're working on now. They're trying to assemble kind of panels in the hopes we can help validate and verify these assays. You need to have these known positives and known samples that have these other coronaviruses, and then to specifically look at that. So we don't know yet, but we're working on it.
Great, and I can see that there are a lot of lab questions coming in for you that we'll get to in a minute.
All right. Just to touch on results, because that's probably most important to you all, we wanted to touch base and share what to do with your results. So if it's positive, we've linked the guidance here, and healthcare providers have 3 options for reporting their laboratory confirmed cases. They can fax, call, or submit an electronic morbidity report, and those are all linked here on the slide. And then we also want to know about your negative. So it kind of depends on where your testing is performed. So if it's conducted at any of the laboratories or facilities listed on the slide, then you don't need to do anything else. But if it's not at one of these places, then we still need to know about it, and it still needs to be reported. And where do you do that? You do it at the number on the screen. So if the lab wasn't on that list, or if you didn't use a point of care test, then it can be faxed to the number on the screen. One thing I wanted to touch base on is this point of care test. That includes things like the Abbott ID Now. So if you're doing that test in-house and not in a laboratory, that also needs to be reported, both positive and negative, and that also applies to any serological tests you're doing. We want to know the results of that as well. So having the negative results helps give us that denominator in all our calculations.
Okay, so another question for you all is what your PPE situation is looking like. So on a scale of 1 to 5, going from PPE, what's that? We have no PPE, to I don't need PPE because I don't do this stuff, or we're swimming in it. It looks like right now we don't have anybody swimming in PPE. Lots of folks that are reusing or have no PPE, and the majority of you that are being very, very careful to conserve what little you have. And then a few folks that say that they actually have enough, which is nice to hear. So we know that this has been an ongoing concern for folks to be able to get PPE, and this might be your answer. So this website that I'm showing you right now, https://arcg.is/1 capital L little I capital CCP and the passcode 8362. That is the direct link to request PPE of any type from TEMA. You can go onto that site, tell them what kind of organization you are, and interestingly, they do not have things like pediatrics, internal medicine, and family practice listed as site types. They have nursing homes and public health departments, so you can click other and type in your organization type if you don't find it listed there, and the PPE will be shipped directly to the address that you provide. I don't think this is a coincidence. I actually entered into this survey yesterday a request for my former practice in Franklin to get 10 N95 masks, and they arrived today. So it's quite possible that if you enter a request into this, you will have your need quickly met. I would love to hear feedback about that and how quickly it's taking to get these requests fulfilled. Please don't put in a request that's asking to hoard supplies. Just ask for what you need to get through the next 1 to 2 weeks, and then they do ask you to go back into that survey every week and report your burn rate so that we can have a better idea of the state as to how quickly these materials are being burned through. But hopefully, if you go to this link and ask for supplies, you will get a box FedExed to your office within the next couple of weeks. So this slide, what frustrates you the most right now? We've made this little word cloud. So testing, uncertainty, changes, lack of testing, people not staying at home. These are the big things that seem to be on your minds right now that are popping up here. Misinformation, yes, and uncertainty, lack of testing. Testing's just kind of staying right there in the middle there. So that's really interesting to see all that. We share all of those concerns with you here too. And then what do you want to hear about next week? So we're really trying to make these webinars meet your needs. So you had said loud and clear that you wanted information about testing and about therapeutics, and so hopefully that answered some of your questions with what we know of right now, and the information that we're giving you is what we're aware of. But feel free to pop in any other questions that you might have about things that you want to hear about on next week's webinar. We do try to address those, and we do read your comments here. So next we're going to go to your questions, and so you can enter your questions in the chat box for the webinar. We'll try to get to as many as we can. We usually get 40 or 50 questions per webinar here, so we'll see what we can get through. The first one is, if a clinic opts to help with testing, does the government provide masks and PPE for staff involved with testing? So not because you're helping with testing, no, but you can request your gear through that TEMA link that I showed a couple slides ago. Does it link with web mobile sign up for patients and appointment slots to prevent traffic backlog? So I'm assuming with the drive-up testing, are there appointment slots? No, there's not. There is no appointment slot sign up. It is first come, first serve for that. Are there any facilities thinking about doing pre-op testing prior to elective procedures? I have heard of pre-op testing in some facilities where they're doing a rapid antibody test And then if that antibody test is positive, then they're going and doing a PCR test before they do their operation. I think you can debate both sides of the logic around that, but that's the one thing I've heard. Have you heard anything else?
I haven't.
And then it looks like this is a good one for Dr. Levinson.
Sure. I'll take this one. The question is, there are so many rapid tests. How does the state view them? What is the best modality for testing, antibody, et cetera? I agree with you. There are a lot of rapid tests out there. It's hard for anyone really to keep up. And really what we would recommend at this point is we are reviewing them as we see them, but we certainly can't keep track of all of them. If you're looking for some guidance, I would start with the FDA. They have a really good website that's frequently updated. If you look up FDA, FAQ, and diagnostic testing, it'll take you right there. There they list all the tests, both molecular and serological, that have EUA authorization. And you can sort by test. So if you sort by serological, it'll show you all 4 of the tests that currently have the FDA EUA. I would start there if you're considering serological testing, and the same goes for the molecular tests, that those are the ones that have at least been reviewed by the FDA, have some basic performance characteristics shown, validated, that kind of thing. So if you're looking on where to start with testing, I would start there. In terms of modality and kind of workflow, that kind of thing, really want to caution folks, especially with serological testing, it's not to be used as the sole basis for diagnosis. So if you're treating patients or considering patients for COVID-19 testing, I would really recommend these molecular testings that look for active virus to see if someone's actively infected. These serological tests are really more for looking at exposure later on, but we know there's a lot of caveats to the interpretations of those. People can be negative today and positive tomorrow. There's certainly a delay from when someone is infected to when you detect antibodies. So we really, really caution the use of that in kind of diagnostic setting at this point.
And at this point, there are no serologic tests with an EUA that are point of care, is that right? Point of care in that without a CLIA moderate to high complexity lab?
That's my understanding. Like I said, it changes by the hour, but all 4 of the serological tests that have the EUAs, I think, all have to be performed in a moderate to high complexity lab. So generally speaking, yes.
But there are some molecular tests that
there are considered waived and can be utilized in the point of care area.
Great. Great. Looks like next one's for you too.
All right, making good quality antibody point of care tests. My understanding is the only validated antibody test is Celix, which is currently on back order from China. Any tests in the U.S. currently have not been validated. Is this correct? Celix is one of the antibody tests that have received the FDA's EUA. There's also these 3 other tests now that are at least given the authorization from FDA. I don't know about their availability. Typically, as these tests get their EUAs, everyone wants them, so I'm not surprised that these are on back order. The most recent one, which is Mount Sinai, that showed up today, I saw their EUA, they do an ELISA-based test, and they're at least based in the United States, so that's something worth looking at. I don't know how accessible or available that would be to folks in Tennessee. But again, I would route people to that EUA list. That's probably the best place to start if you're trying to figure out what the good quality tests are. Those have at least been reviewed by FDA, and those are your best bets.
So for those of us who are not laboratorians, the Mount Sinai test, this is a test that Mount Sinai is running, not one that they're selling.
I believe so, yes. It came out today, so I've known about it for about an hour.
All right, saliva tests.
Yeah, so there's one, this came out of Rutgers that just got their FDA EUA this week. They have a saliva collection device. I know a number of other places are also looking at saliva as a specimen type and comparing it to other specimen types like nasopharyngeal or oropharyngeal. There's certainly work, and people are already doing self-collection, especially for nasal swabs. So these are all in process. There's studies that are just beginning to get published now kind of comparing the sensitivity of these different specimen types. So it's very much a moving target. I wish we had clear answers, but those studies take time to do, and we're all in this together and figuring it out as we go. I would say that while we are looking at different specimen types, it's important to keep track of where you're sending your testing and the lab and what their acceptable specimen types are. So even if saliva is an acceptable one somewhere, you need to check with your lab and make sure that's one of theirs, because different assays accept different specimen types, and you want to make sure you don't get it rejected. So
it's the one advantage to something like a saliva test is that it would help with PPE burn, because you wouldn't have a healthcare worker necessarily having to be with them during an aerosolizing procedure.
Certainly, that is definitely one advantage. I think the concern is a decreased sensitivity, that you may have less detectable virus there compared to these other specimen types. So that's what we're working, but it also depends on the stage of infection that someone might be at, whether or not they're even symptomatic. There's a lot of unknowns that we're still trying to piece together. All right, I will take the next question. It says, the FDA has authorized a new manufactured self-test swab. Considering the probability of bad swabbing, how will that impact your curve results? It certainly will. I'm not sure if we can really track how it will. So, as I mentioned at the beginning when I started talking on my slides, that really the quality of the laboratory result is based on, largely on the quality of the specimen that we get. So certainly bad swapping is certainly an issue, kind of a garbage in, garbage out. So if we get a poorly collected sample, then we have less weight that we can put on that result. And sometimes it's hard to know, because in the laboratory, we just get the samples. We don't know how they're collected necessarily. So I share your concern. It certainly could impact the curve and the results, but these are all things that are being examined right now.
And you want to speak to sensitivity and specificity?
Sure. I can give you the quick answer. We don't know yet. And that's true for both the rapid antibody tests and for a lot of the molecular tests. There is data, things like limit of detection. It depends on your assay. And there's some limited data that they put generally in the performance characteristics of these, but you really have to be careful in how you interpret that and what they're compared to, what that gold standard is, whether or not these were spiked samples from asymptomatic or hospitalized patients versus asymptomatic patients. You really have to be careful in how much you interpret those. So these broader studies to really evaluate the sensitivity and specificity of a given assay and then of these assays against each other is ongoing.
All right, the next one is, given that Plaquenil's hydroxychloroquine is very safe with or without azithromycin, and many internists have used it, what is the status at this time of our state for physicians using this upfront in treatment until something better? So, I don't think hydroxychloroquine is generally considered very safe. It's a medication that has been used commonly in people with conditions like rheumatoid arthritis and lupus, where they're monitored and where they have conditions that are life impairing. But it is still not approved by CDC or FDA as a medication that has been shown to be safe or effective in the treatment of COVID-19. So at this point, I think there are some institutions where if they've got someone in their ICU and they've run out of options and they want to give hydroxychloroquine, that's something that is certainly up to their discretion. But as far as our giving a recommendation to give this out to the masses to either prevent or treat COVID-19, I don't see that coming very soon. Any update on staff who have tested positive and are asymptomatic? We are removing from work and they are quarantined and retested. Concerned is hearing there is discussion allowing them to provide care to residents who are positive. So I've not heard that discussion, but it seems reasonable if we're cohorting patients who are COVID positive, that them having a COVID positive provider might be something to at least discuss. As far as healthcare staff who are tested positive and asymptomatic, they should not be providing clinical care while they're positive. The CDC has reverted back to its recommendation that healthcare workers have 2 negative PCR tests spaced 24 hours apart before they return to work, and if for some reason they are unable to be tested, then they can go with the 7 days in isolation plus 72 hours of return to wellness. But they have gone back to recommending that healthcare workers have 2 negative tests before they return to patient care. Someone's asking for the TEMA website, and we will get these slides posted. Peter, will we have them up today, do you think? Today or Monday? We can, I can shoot back and put that, there we go. There, now it's on the screen. Okay, yeah, go ahead.
I can take the next one if you'd like, because it gets back to testing. The question is, help me understand what providers do with serological information. That's something we're currently working on, is kind of trying to establish what a workflow or testing algorithm would be for both molecular testing and serological testing, whether you would do one and then reflex to the other based on the result. Right now, we really caution providers with interpreting that serological data for anything in relation to diagnostics. So really, you want to be using these PCR-based tests to diagnose your patients with COVID-19, these serological tests. We really just don't know enough. A positive could be due to cross-reactivity with another human coronavirus. A negative could mean that they just haven't mounted detectable antibodies yet. We also know, even if it's positive, we don't know if they're protected necessarily. So there's a lot we don't know. We don't know how to interpret those serological tests yet. So until we have more information to guide that, we would really push providers to using molecular tests for diagnostics.
All right, the next one is, can we expect more drive-up testing, and will the health department go to rapid testing so people can get results before they leave? So most of the health departments right now have drive-up testing Monday through Friday. I encourage you to call ahead to find out what their hours are for doing that, but most of them are equipped to doing that. And then this weekend, they have these surge events in over Saturday and Sunday in those 33 different locations that we put up that are also available on the tn.gov website. If you go to the red bar at the top of the screen and then go to remote assessment sites, there's a list of those sites for this weekend where that testing can happen. We won't be going to rapid tests at the health departments anytime soon, I don't think, for a number of different reasons. One is just availability of that test. The one Abbott test that's been in pretty low availability. There's a few clinics in the state that have those right now. There's cost associated with that, and then even just having the backup of 45 minutes waiting for your test results while we're trying to do drive-through testing probably makes that prohibitive for that to happen. But the turnaround time has been much better recently. Of course, that depends on the number of specimens that are being put through the lab at the time, but generally those results will come back within 48 hours or so, 48 to 72. It is the recommendation that if you get a test that you isolate yourself until those test results come back. So this one's about the Abbott Rapid PCR test that's available at American Family Urgent Care Centers. So the last I heard, it was still available in Spring Hill, but they had a limited number of tests there. And I have not yet heard if they've been able to move those across their other sites in the state. Those tests are not free. They're $150 at the point of care. And you do have to call for an appointment to get those tests done. If you do not have insurance, then the provider could bill the state for $70 worth of that test, but I imagine that an uninsured patient would be responsible for the balance. Are you recommending that asymptomatic people get tested to get an idea of general population rate, or only if someone suspects they've been exposed? So that's a really hard question and I think there's debate on both sides. So a negative test is a negative test today, like Dr. Levinson said. So if you get a PCR test today, with or without symptoms, and it's negative, that doesn't mean that you're not COVID or coronavirus positive tomorrow. It doesn't mean that you weren't coronavirus positive yesterday. It just means that today coronavirus RNA was not detected. And that not detected is also very dependent on the way that that swab was obtained, whether or not there's significant viral load at the time, whether or not there are symptoms. There are so many variables. PCR tests generally are not falsely positive for the most part.
Yeah, that is, just to chime in, there is one thing. Most of these molecular tests that have gotten their EUA, they've at least looked at cross-reactivity with other viruses, including human coronaviruses. So,
yeah. So a positive is probably a positive, more likely. We're more confident in a positive PCR test than a negative PCR test, I think it's probably fair to say.
Yeah.
So if you are asymptomatic and you have a positive PCR test, then the recommendation is that you would isolate for 7 days. Well, if you never had any symptoms to start that clock, then that 7 days goes from the day of your test until you have 72 hours of. So it's possible that you could get tested today, be positive today, not develop symptoms for 3 more days. Then you have to clock from 7 days from that point for your isolation. So now you're in for 10 days, and then you have to be at least 72 hours well. So that's kind of how that goes. So yes, testing too soon can give a false negative, and you can eventually end up with a positive. We can also have people who are very, very symptomatic who would meet a clinical diagnosis of a probable case of COVID-19, which is someone with a fever and cough, who has a negative PCR test. And we should still treat those people as if they're infectious for the time of their illness. So 7 days from the onset of their symptoms, plus 72 hours of wellness if we're going to prevent spread through the community. Are the health departments testing children without comorbidities? Yes, the health department will test anyone who would like to have a test. So there are some institutions that are not testing certain populations, and that is entirely up to them and how they choose to use their lab. But the Department of Health will test anyone regardless of symptoms, regardless of age. And the state public health lab, if a provider wants to call for our authorization to have a test done in state public health lab, if there's a concern about a comorbidity and there is not a commercial lab available to you, then we can run that test through the state as well. Will schools be required to open back in the fall even if we aren't sure about immunity? So school closure is done on a district-by-district decision level. I think right now, at least from what I'm hearing out of folks from DOE, the plan is to open in the fall and to find ways to make that happen. And whether or not that depends on community immunity and our understanding of that, I don't think is clear at this point. But I think everyone feels that it's a priority to try to get children back to school safely and to make whatever accommodations have to be made to allow that to happen, whether that's online learning or staggered starts or distancing or whatever kind of accommodations they need to use. Can you tell us what we can do for alternate PPE when we run out? So the CDC is still recommending the use of things like bandanas or homemade fabric facial coverings. Those can certainly still be used. Different clothing coverings can be used, lab coats that can be laundered, patient gowns that can be laundered. Some sites have used contractor bags as clothing as clothing coverings to be able to help. There are lots of conservation resources available too. If you go to the Emory website, they have some really good conservation resources, as do we at tn.gov/health. Go to the yellow bar and then go down to healthcare providers, and you can find conservation information there as well. And then one last question, since we're at 1 o'clock. Are PCR tests done at the health departments different than the ones that are done at Quest? And is the turnaround time faster with TDH?
I'll answer that one. It's a moving target, and it depends on what test Quest is doing and what the public health lab is doing. So because Quest has a number of sites that are doing testing, and their assays tend to be more high throughput because they get so many more samples, it really depends. I believe they're using an EUA test, so I don't know if it's the same one as what we're offering at the state public health lab. And the turnaround time, it really varies. I believe that's published, or at least available, that the turnaround times, it really depends based on reagents and the number of samples each laboratory gets, whether or not an instrument goes down and there's a backlog. It really varies. I can speak to the State Public Health Lab, and our testing turnaround time is typically 24 to 48 hours, depending on when we receive the sample, but we're generally much closer to that 24-hour mark. So 1 to 2 days, pretty good. It's, I think, one of the faster ones you'll find within the state in terms of laboratories. I can't speak to Quest. These are all moving targets as we kind of work our way through this pandemic. So,
yeah, working our way through, I think, is the key here. So it's uncharted territory for everyone. So I'd like to thank you, Dr. Kara Levinson, for joining us and giving us some really fantastic information about testing. We hope to have all of you tune in again next Friday, and please complete the survey that through Menti so that we can find out what topics you'd like to hear about next week. And in the meantime, stay safe, and we'll see you then.
Thank you.